Hello, this is class Bio 225 with Dr. Lucy Afgharian. We are going to talk about genetics from genes to genomes. Today is chapter 6. We are going to finish chapter 6 today. This is part 3. So let's continue from we were left the topic last time. Pick up from there. So we were talking about how the DNA information is being replicated. So, and then afterwards, we want to make sure that the information that DNA carries is reliable, accurate, and there are three ways to do so. One is redundancy. Either strand of the duplex helix can specify the sequence of the other strand. And then also precision of cellular replication machinery, where DNA polymerase 1 and 3 have proofreading ability, and we were going to talk about this in chapter 7 in more details, where like from 3 to 5 endonuclease it goes back, choose DNA and makes it again. And then the DNA repair enzymes that we were going to describe in chapter 7, and we will talk about this in more details there. Okay, as a review, let's talk, because we talked about this already before, let's talk about like recombination at the DNA level. So during the recombination, the new combinations of alleles are being created by two types of events in meiosis. It's independent assortment, where each pair of homolog chromosomes segregates freely from each other, and we have talked about this when we were going over chapter 4. And also the crossing over, where two homolog chromosomes are being exchanged, are having exchange portions within DNA. And we have talked about this while we were doing chapter 5. So, and it creates new alleles with a combination of linked genes, and it ensures proper chromosome segregation during meiosis. And we are going to talk about mistakes that can result in non-junctions when we go over chapter 13. So how do we know modes of occurrence of recombination? So let's try to understand, is there a way to see that the DNA molecules break and rejoin during recombination? Apparently, there is an experimental evidence, and if you recall 2 classes ago, we have talked about how there was conducted an experiment with tagging molecules with heavy or lighter radio labels. So here, Messelson, Wiggle, they did the same type of experiment where they co-infected E. coli with radio-labeled phage, bacteriophage lambda with genetic markers that had heavy C13 and N15 or light C12 and N14 isotopes, and they looked at the results in cesium chloride density gradient. Detecting the DNA, and of course we know already from the previous class that there is being a differentiation between light atoms and heavy atoms. And here they found out that the genetic recombinants had DNA with hybrid densities, and you can see that in the picture also nicely presented. So heteroduplex regions occur at sites of genetic exchange. What does it mean? So when the DNA is opening, the DNA strands, they don't break and rejoin at the same location. there is a term called for the sites that are being break, they are called sticky ends. So there is a region of DNA that is that has an overlap. So when one part is breaking and another part is breaking on another side, another DNA, they have to have some overlaps in order for the base pairs to come and recognize each other, the complementary pairs, and stick together. That's why they are called sticky ends. So the breakpoints on each strand can be from hundreds or to thousands of base pairs apart. And there is a term called heteroduplex regions of DNA, heteroduplex, which is a region of the DNA that is between the breakpoints. And one strand is maternal. In some cases, for heteroduplexes, one strand can be maternal and another can be paternal, or like strands. Also, the strands can have mismatches, and that you can see on Figure 623. So let's talk about the recombination model. The tetrate analysis in yeast showed that only 2 of 4 chromatids are recombinant. Recombination occurs only between homologous regions and is highly accurate. Also, crossover sites often associate with heteroduplex regions. Gene conversion can occur in absence of crossing over, and not all combinations lead to crossovers. And let's look at that now in more details. So here are the points for meiotic recombination, the double-strand break repair model. So homologous chromosomes break, exchange DNA, and rejoin. Also, breakage and repair create reciprocal products of recombination. And recombination events can occur anywhere along the DNA. And finally, precision in the exchange prevents mutations from occurring. And if you can recall, precision in the exchange is also basically one of the reasons that we were saying that we can be sure in fidelity of DNA information. Now let's go step by step through double-strand break formation for model of recombination. So DMC1, it's a enzyme that breaks phosphodiester bonds of both strands of one chromatid. SPO11 in yeast would be the homologous to DMC1 of multicellular eukaryotes. So here you can see that the DMC1 comes and breaks the DNA, breaks the phosphodiester bonds there. And then 5 ends of each broken strand are being degraded to create 3 single-stranded tails. That's step 2. And in step 3, one single-strand tail invades a non-sister chromatid and forms stable heteroduplex. In this picture, you can see the DMC enzyme in orange, and the yellow ones are the single-strand binding proteins that are coming to stabilize the new structure. Step 4 is called formation of double holiday junctions, where D-loop enlarged by new DNA synthesis at 3 end of invading strand, and all the new DNA synthesis is going to come to fill the gap in bottom strand using the displaced strand as a template. Branch migration. This is how step 5 is called, where the heteroduplex region of both DNA molecules is lengthened. Step 6 of the model of recombination is called holiday intermediate. During this step, there is rotation taking place of two chromatids. They rotate by 180 degrees. This is something that we have to imagine, like in 3D, basically. Step 7, it's called alternative resolutions. And here cutting is taking place, and cutting of holiday junctions by endonucleases is equally likely in either vertical or horizontal plane. And step 8, the probability of crossover occurring. So now, remembering the previous slide where the cut can happen horizontally or vertically. So it can either resolve in non-crossover occurrence. That will happen when both junctions are resolved in same plane, or we can have crossover, which will be the case when two junctions are resolved in different planes. So for example, when we have horizontal, we are not going to have crossover. When we have vertical, we will not have crossover. But when we have horizontal vertical or vertical horizontal, that's when we are going to see the crossing over.